Friends, welcome to Biology Made Easy. Today in this video, we'll discuss Mendel's experimental techniques. In earlier videos, we have discussed how many characters was taken by Mendel, what are the 7 pairs of contrasting characters, where they are located, what are the regions of his success, but still why he was not recognized. All these things we have discussed. But what is the method he is working? What are the techniques he adopted? That is also important. So in this video, we'll discuss his experimental techniques. His experimental techniques we can discuss in 4 headings. The first heading is selection of pure parents. Selection of pure parents. Now, as already we have discussed in the earlier video, initially Mendel took 34 pairs of characters. Initially took 34 pairs. Then he reduced it to 22 pairs. And finally, the 7 pairs of characters. And what are the 7 pairs of contrasting characters? That already we have discussed. Now, in the 7 pairs of contrasting characters, they are present in 14 plants. Because suppose we will take a tall and dwarf one contrasting character, that means one plant will be tall, another plant will be dwarf. Suppose you take a round or wrinkled seeds, one plant will have round seeds, the other will have wrinkled seeds. So for 7 pairs of characters, there will be 14 varieties. But these 14 varieties should be pure breeding. It should be true breeding. There should be pure lines. How can we determine it? So that is determined only by means of selfing for 4 to 5 generations. Suppose we'll take a tall plant. We do not know that whether this tall plant is homozygous tall or it is heterozygous tall. If by chance it is homozygous tall, then there is no problem. We will get our desired results. But suppose this plant is heterozygous tall, because phenotypically it is looking tall, we cannot see what is the genotype inside. So if this plant is taken, the entire results will go wrong. That is why before conducting any experiment, the plant is to be breeded or to be bred for several generations, for 4 to 5 generations. So when this will be bred for next generation, it may give rise to again tall and dwarf plants. Then again it may give rise to tall and dwarf plants. And finally, after several generations, it will give rise to only tall plants, and then only will be confirmed that the plants are pure line, they are true breeding, they are homozygous state. Then only we can take that plant for our experiment. So that was done by Mendel. He selected the pure lines. Then the second step. The second step is hybridization. So what is hybridization? Hybridization is artificial crossing between 2 plants. And you know that the pea plant is predominantly self-pollinated. So if we leave the flowers as such, it will be self-pollinated. But Mendel conducted hybridization between 2 different plants having 2 different contrasting characters. So in this case, what is to be done? One plant will be treated as female plant, the other plant has to be taken as male plant. Suppose the tall plant will act as female plant, then in that case, the dwarf plant will act as male plant. And the result, suppose it came as a tall plant. But suppose it will be reverse, suppose this will be female plant, this will be male plant, then also Mendel found that the result is same. That means he made a reciprocal process for his hybridization. So when a reciprocal process, that means what? That means suppose in this tall plant there are 100 flowers. Suppose here also there are 100 flowers. Suppose. That means out of this 100 flowers, 50 flowers they were female and the other 50 flowers male. Similarly, in this case, 50 flowers female and 50 flowers male. That means when these 50 flowers are acting as female flowers, then from the pollen grains of these male flowers are dusted away. And when these 50, the same plant, when these 50 female flowers will be pollinated, then the pollen grain of this will be dusted. That means in both the plants we will get seeds. This is known as reciprocal cross. So for his experiments he made the reciprocal crosses. And he found that the results are same in both cases. Now when we made hybridization, the hybridization has several steps. So the first step of hybridization is emasculation. Now what is the meaning of emasculation? Emasculation means removal of anthers from the female plant or making it sterile. When we are taking these flowers as female flowers, because the flowers are bisexual. So when these are taken as female flowers, that means the pollen grains, the anthers of these flowers are not required. That means that from these 50 female flowers, the anthers are to be removed. That is known as emasculation. When these 50 flowers will act as female flowers, the anthers will be removed from them. That is, emasculation means removal of anthers or making them sterile. In which flower? Female flowers. Because in female flowers, the anthers pollen are not required. So to maintain purity, the anthers are removed. That is known as emasculation. And this is usually done in the female flower. So either it can be done by hand, by means of forceps, or by alcohol, or a hot water, cold water, or there are different chemicals, but emasculation can be done. That is the first point. Then the second point is bagging. Now what is the meaning of bagging? Suppose you have removed the anthers from a flower and you want to pollinate it with the pollen grains of a different plant. But when you will emasculate, the flower is to open. But if you leave it, then the foreign pollen grains may come and they may fall on the stigma of this plant. And when you will make cross, already it is pollinated. That is why immediately after emasculation, there is a bag to be put over the flower so that it is not contaminated by any foreign pollens. That is known as bagging. Then the next point is crushing. Now you have put the bag. Now the next day you have to make a crush with the desired male plant. Suppose this plant is acting as a male plant. So you take, in this case also the flower should be bagged so that there should not be any contamination. So on the day of crushing, you just remove the bag from the female flower, remove the bag from the male flower of this plant, collect the pollen grains and dust it on the stigma of this plant. That is known as crushing with the desired plant. And after crushing, again you put the bag so that there will be no contamination. And the next point is tagging. Now what is tagging? In the same plant, 50% of flowers are female, 50 flowers are male. So which one is female and with which plant you have crushed, how will you maintain that record? You can put the record in a notebook in black and white, but in the plant how will you identify it? That is why a tag is put in the pedicel with which plant on which date it is crushed. That is known as tagging. Then after a few days you find that within that bag the fruit is developing. That means the crush is successful. And when the fruit is completely ripened, then you can harvest the seeds. Harvest the seeds. Now the seeds of these fruits, they are known as hybrid seeds. Hybrid seeds. Now these hybrid seeds when grown for the next generation, that gives rise to the first filial generation or F1 generation. So the next point is selfing F1 hybrids. Now when these hybrid seeds are grown, they will give rise to plants which are known as F1 hybrids. That means first filial generation, first generation. So usually the hybrids are heterozygous, and usually they are sterile. Though in case of pea plant they are fertile, but in most of the hybrids they are usually sterile, and they are usually not stable. So to bring stability, to make it more homozygous, usually the F1 hybrid is self-pollinated for another 2 to 3 generations, like F2, F3, F4, like that, several generations, so that the plants will be stable. So this is known as selfing of the plants. And sometimes here also back process are made, because the desired character should be present in more. That is why if you cross it with the desired parent, then that character will be added. That we'll discuss what is the backcross, test cross, we'll discuss later on. So sometimes the F1 hybrid is backcrossed, and then again it is self-pollinated. Then come to the fourth point. The fourth point is calculations. Now after carrying out all these experiments, Mendel calculated them, which are difficult to accept, to be accepted by other scientists. Because during calculations, he applied the theory of a probability, and algebraic formulae. So here what he did, he applied the binomial expression, that is ax plus by whole square, where a and b are alleles in equal proportion. Now in that case it will be x square 2xy plus y square. Now if this will be turned to the digits, then it will be half square, then 2 into 1 by 2 into 1 by 2 plus again 1 square. So this is giving the result 1 by 4 plus 1 by 2 plus 1 by 4. That means we can tell 25%, 50%, and again 25%. Why he applied this? Because when there are two alleles, they give rise to 3 types of plants. Suppose there are this is the heterozygous plant, and these are the 2 alleles. How many types of gametes it will give rise to? It will, in the male side it will give rise to 2 types, and in female side it will give rise to 2 types. So if they can cross in all probabilities, then this will be capital T, it is tall plant, capital T, small t, tall plant, capital T, small t, tall plant, and small t, this is dwarf plant. So you see, the tall plant, this is coming in 25%, but this is homologous tall. Then this is homologous tall. Then this is heterozygous tall. This is capital T, small t. They are coming in 50%. This is 25%. That is 50%, because 2 are there. Out of 4, 2 are there. And this 25% is homozygous dwarf. This is small t. This is also 25%. So he applied this calculations to his experiments. So these are the different steps or his experimental techniques which he applied. So thank you very much for watching the video. God bless you. Have a good day.